Hi, and welcome to segment 4 of 4 of the lecture topic called Cells and Movement Across the Plasma Membrane. Today we will wrap up this large lecture topic. I'll describe cell junctions and the extracellular matrix, and those are important for understanding tissues. And then I'll talk about gradients in the universe and how they apply to physiological systems. And so if you're using the PDF slide presentation, today I'll be covering slides 133 through 144. So cell junctions. I hope you remember that a tissue consists of a group of cells that work together to perform a common function. For most tissues in the body, those cells are connected to each other, and they're also connected to structures in the extracellular environment. So this diagram that comes out of a textbook, maybe your textbook, this diagram shows 5 different types of cell junctions. Some of them connect that central cell to its neighbors, and one of them down below, D, you can see that cell junction D is connecting the cell to an extracellular material known as the basement membrane. And so cell junctions come up in the context of the plasma membrane because cell junctions include, their structural components include proteins that are part of the plasma membrane. I'm going to tell you about 2 types of cell junctions. Shown here, letter A is a tight junction and letter E is a gap junction. Of course, all of the junctions are important, but tight junctions and gap junctions come up for us down the road. So tissues, different types of tissues in the body have different physical characteristics. And you can think about tissues in terms of fabric and the different characteristics that a fabric might have. So compare spandex, which is really durable and stretchy, to tissue paper. Spandex would be analogous to the tissue that makes up the dermis of your skin. Your skin is pretty durable and stretchy, and that's dense, irregular connective tissue. Whereas tissue paper, and not just tissue paper, but wet tissue paper, is analogous to the delicate lining of the air sacs in your lungs, that simple squamous epithelial tissue. So for any given tissue, the cells that make up that tissue, how they are connected to each other, how they're connected to their extracellular material, all lend to the physical characteristics of any particular tissue. So tight junctions. Tight junctions are often described as a quilting or a stitching together of the plasma membranes of 2 neighboring cells. And the purpose of tight junctions is to prevent material from leaking or passing in between the 2 cells. So one place that you have a lot of tight junctions is in the lining of your small intestine. And we can imagine the panel that's on the left here, we can imagine that those cells are forming the lining of your small intestine. So above those cells, we have, a mixture of digested, partially digested food material. And in that food material, there are bacteria. And we don't want those bacteria, which are tiny. They're approximately 1/10 of the size of an average human cell. We don't want those bacteria to be able to pass through those neighboring cells and somehow get into our blood and cause an infection. So our example tissue, the simple columnar epithelial tissue that's lining your small intestine, those neighboring cells are joined by tight junctions. And the second cell junction that we'll cover for right now are gap junctions. And gap junctions also connect the plasma membranes of 2 neighboring cells, but they have a much different structure and a much different function. Gap junctions physically join the cytosol of the neighboring cells. They form little tunnels or culverts, passageways for fluid and small particles like ions to easily move between the cytoplasm of one cell and the cytoplasm of the neighboring cell. So gap junctions will come up for us when we talk about, when we talk about cardiac muscle tissue. The movement of ions that precedes muscle contraction, those ions need to move, and in cardiac muscle tissue, those ions are able to move between one muscle cell and the neighboring muscle cell by passing through these gap junctions. And then, as I mentioned earlier, cells that make up a tissue, they may also be connected to materials in their extracellular environment. And that extracellular environment is often a mixture of water, solutes, and solutes including proteins. And that extracellular matrix also lends characteristics to the physical properties of a tissue. So for example, think about the physical properties of bone versus the physical properties of hyaline cartilage. Hyaline cartilage makes up the squishy part of your nose. And obviously bone is hard and solid like a rock. And that's due to mostly the differences in the composition of the extracellular matrix. And so the extracellular matrix surrounds cells in a tissue sometimes, and we'll usually just refer to it as interstitial fluid or extracellular fluid, but I do want to draw your attention to the fact that it is likely a matrix. There's fluid and proteins there, and the cells may or may not be directly linked to the proteins. And this brings us to our last topic for this lecture segment, and it's a big one. It applies to the entire universe, not just the human body. And here's a great sentence. Nature abhors a gradient. Do you know what it means to abhor? It's an unusual vocabulary word. It's like a SAT word. To abhor means to hate or to despise. So nature hates a gradient. Nature, and I mean throughout the entire universe. So across an area that is larger than any of us can comprehend. Nature is constantly giving rise to gradients and undoing gradients, erasing them, making them go away. So a gradient most generally can be defined as a difference over a distance. So we have been talking about concentration gradients a lot recently. We talked about concentration gradients as being important for diffusion, both simple diffusion and facilitated diffusion. Concentration gradients drive osmosis. And we talked about how cells can use active transport to create and maintain a concentration gradient. You can think about mechanisms of passive transport as trying to undo a concentration gradient, right? When we reach equilibrium, there may not be a concentration gradient left. And active transport requires the cell to spend energy to create a concentration gradient, or at least maintain that concentration gradient. We can also envision a temperature gradient. So you have what's known as a shell body temperature and a core body temperature. Your core body temperature is generally warmer than your shell body temperature. This is why it's possible for you to walk around with a body temperature of roughly 37 degrees Celsius, but maybe your hands are cold, or your skin feels cold, or your nose is cold. Because closer to the surface of your body, especially if you're in a cool environment, closer to the surface of your body, there's more dissipation of heat into the atmosphere. But deeper in your body, towards your body's interior, there is a stable warm body temperature. So as we move from your deepest viscera out toward the surface of your body, we will observe a temperature gradient. And then there are also pressure gradients. Pressure gradients will come up for us when we talk about the respiratory system. We'll specifically talk about air pressure gradients, because air will always flow from an area of high pressure to an area of low pressure. And perhaps you can imagine the meteorologist, the weather person on the news talking about a low pressure system. Pressure gradients in the atmosphere drive the flow of air, and we call that wind. Pressure gradients that our respiratory systems generate are what are responsible for air moving into and out of our lungs, just like wind in the atmosphere. All right, so an important thing to recognize about gradients is stated there on the slide: the steeper the gradient, the faster the rate of its undoing. I want to show you some examples of gradients that really don't directly have anything to do with even biology, never mind human physiology. So these gradients would exist even if life did not exist on planet Earth. These gradients would still be there. So what we're looking at here is the movement of the tectonic plates around the planet. And the movement of these tectonic plates that form continents, and over geologic time, continents will break apart and new plates will collide into each other and create a mountain range, etc. So maybe you've learned about this in another science class. The movement of these giant tectonic plates is driven by temperature gradients in the mantle of the Earth. So in the liquidy rock part of the Earth, that liquidy rock material, obviously it's molten rock, so it's super hot, but even within that molten rock, hot molten rock rises and cooler molten rock sinks to the bottom or toward the core of the Earth. And those currents that are in the mantle of the Earth are driving the movement of the Earth's crust in these tectonic plates. Again, here's an example on planet Earth of gradients that exist whether or not life exists on the planet. Earth would have oceans, and water circulates in mass quantities around an individual ocean and in between oceans. And the movement of that water is driven by temperature gradients. Warm water will rise and cold water will sink. And salinity gradients. Salt water is denser than less salt water. And so that gives rise to these gyres, or these large currents that drive the movement of water around the planet. And it's important that this water also carries heat around the planet. So this movement of water is also has a huge influence on climate and weather. Look familiar? We've talked about this image before. Obviously, living things need to exist to create this dam, right? It's a human-generated substance. The purpose of this dam is to create what's known as a gravitational potential energy gradient. Water wants to move downhill. The dam prevents it from moving downhill. But humans have the capacity to open up the dam and allow that water to move downhill along its potential energy gradient. And as potential energy is converted to kinetic energy during the movement of water, humans are so clever that they can harness that energy to generate electricity in a hydroelectric power plant. So why is this property of the universe, that nature hates a gradient, why is that important in your physiology class? Because all forms of life have been shaped by this force of nature. Every single form of life has evolved with this condition in place, that gradients exist and nature is constantly trying to undo them. And living things have evolved to harness the power of gradients. For example, how do we move air into and out of your respiratory system? Well, natural selection came up with a way. Hey, let's generate a gradient, and that will cause the air to flow. And then as we've started to see, living things use ATP to create gradients or to overcome them. And so far, we've seen ATP being used to create a gradient with our exploration of the sodium-potassium pump. So just to remind you, here's a simple concentration gradient. We've looked at that before. Here are two different concentration gradients, a gradient for purple dots and a gradient for orange dots. And this is a passive process. By simple diffusion, both the purple dots and the orange dots will diffuse along their concentration gradients until equilibrium is achieved. And at equilibrium, there are no more concentration gradients. Nature hates a gradient. Nature just undid two gradients by simple diffusion. And here's another example of a gradient, but it's a gradient that is useful. For one reason or another, the cell creates this gradient by spending energy. This is a process known as active transport. We're creating a proton gradient, also known as a hydrogen ion gradient. This will come up later when we get to our mitochondria. Our mitochondria make most of our ATP, and part of that process involves a very steep hydrogen gradient inside of them. So that's a later topic. And then you remember gradients also drive the movement of water by osmosis. And we recently talked about electrochemical gradients. So not only do concentration gradients, but electrical charge gradients influence the rate and direction of the movement of a substance across a membrane. And a membrane potential is a type of gradient. There's potential energy in membrane potential. Positive and negative charges are attracted to each other. They want to move across that gradient, just like water wants to move down the dam, right? But the plasma membrane, that phospholipid bilayer, keeps those positive and negative charges separated. And they can only move once we open the dam, so to speak. So an ion channel has to open to allow those charged particles, those ions, to cross. And as we'll see very soon, that's how we generate electricity in our cells, physiological electric currents. So looking ahead, I hope you have an understanding that every physiological system relies on one or more gradients. And so far we've come across some of these: concentration gradients, electrochemical gradients, osmotic gradients we've definitely talked about, and looking ahead, we will eventually include pressure and temperature gradients. So that brings us to the end of segment 4 of the lecture topic, cells and movements across the plasma membrane. And here's a list of all of those lecture topics that we've talked about over the 4 segments. Thanks for listening. Bye-bye.